I am the action potential. I'm causing a depolarization in the axon of the nerve.
I am calcium. I'm green.
What are you doing, calcium?
I am pushing the synaptic vesicle.
I am the vesicle carrying acetylcholine. I am now passing through the synaptic membrane.
Pushy, pushy.
I am now releasing the acetylcholine.
Glitter?
I am now acetylcholine opening the motor end plate for
Sodium.
And potassium. I'm an action potential, heading down the sarcolemma to a T-tubule.
Woo!
Say action whenever.
Action.
This is the whole muscle, and inside the muscle are mitochondria, and those are like the green things. Okay? And then inside the whole muscle are fascicles, which are bundles of fibers. And inside the fascicle are fibers, and inside the fibers are myofibril.
And the myofibrils are made of mitochondria. They're not made of mitochondria. They're made of myosin and actin. And the myosin head would be attached to an active site on the actin. And then the ATP molecule, which fell on the floor, would float by, bloop, and attach itself to the myosin head and release it. And then the ATP energy would go into the myosin head and then would turn into ADP. And then it would cock the head and put it in the active and ready position. And when calcium floats into the muscle fiber, it would bind to the troponin, pull the tryptomyosin off of the active site, exposing the active site, and then the myosin head would be irresistibly drawn to the active site, leaping forward, attaching to the actin molecule, and dragging it along, bringing the Z disc along with it toward the M line. And both of these things would be happening on either side of the M line, and thus causing the sarcolemma to shorten and causing muscle contraction.
Go.
This is the sarcoplasmic reticulum, which would be over the entire myofiber, and right here we would have transverse tubules, which would be the yellow ones, and this blue one would be the terminal cisternae, and that would have the calsequestrin, which would basically draw the calcium into the sarcoplasmic reticulum. And then here we have a representation of the triad, which would be 2 terminal cisternae and 1 T-tubule in the middle. And then again we'd have the sarcoplasmic reticulum, which is part of the terminal cisternae, in between, and then again we'd have another triad, and then it would repeat throughout the entire length of the muscle cell.
This is acetylcholine, and to break acetylcholine down, you have to get acetylcholine esterase. An esterase breaks acetylcholine down into choline and acetic acid.